Well, good morning, everybody, and I don't know when you all arrived, but welcome back to Old Nassau. And I guess, this is a big weekend. Like, you've got visits here today. I guess there's, you're gonna learn about Ukraine and Russia, right? And then there's the Davidson game. So this is another stuff that I may have missed. What's that? Yeah.
I grew up at Davidson.
Okay, well, a very fine school. A very fine school doomed to lose tomorrow. So here's what I want to do today. The schedule says that I am up for an hour and 15 minutes, and I assure you that I cannot talk for that long. So here's what I wanted to do today. I wanted to talk about neuroscience. By the way, welcome to our new building. We moved in January. We're super happy about this building. The psychology department moved down from Nassau Hall. Some of you may have been psych majors. Not Nassau Hall, Green Hall, yes. Well, actually, Debbie Prentice, chair of psychology, moved up to Nassau Hall. Anyway, but psychology moved down from Green Hall. We moved from molecular biology. Several of us moved down from the molecular biology buildings, and there are people from computer science. And so we're all finally together under one roof, and I'm really super happy to welcome you to our new building. This is, before it was ever built, this is an artist's rendering of it. And this is, as you can see here, there was something called value engineering. So if this doesn't look like the shape of the psychology building, that's because value engineering has made it nice and square instead of this rounded shape that you see here. Let's see. So anyway, welcome to our building. What I want to do today is I want to talk about neuroscience, and I want to talk about where neuroscience may be headed in the coming decades, where Princeton may make a contribution. And I want to turn to a topic that's of great interest to me, which is child brain development, and also specifically autism, and also human individuality. So I'm going to be spanning a pretty wide range of topics. And part of my reason for doing that is to introduce you to what's happening here at Princeton, but also part is to spur a discussion, because what I want is to not go on for too long and to start a question and answer and a discussion to find out what you all are interested in. As you heard from the title of my book, I also have written for general audiences about things like aging and exercise and all kinds of things. And so there's a pretty wide range of topics that I would be delighted to talk about. So first, let me just say that neuroscience obviously is one of the great frontier disciplines in science today. I was a physics major when I was an undergraduate, and I left physics to go into neuroscience. And my motivation for doing that was that physics was a great discipline where one could learn about deep questions. But I noticed, even when I was an undergrad in the 1980s, that physics was getting very mature. And the average time between discoveries was getting kind of long. And I just thought, actuarially, based on my own life expectancy, that it might be nice to make a contribution to whatever field I entered. And so I picked neuroscience, and I've been very pleased with that choice. Now, here at Princeton, we have different kinds of strengths. One is in cognitive neuroscience, and I think you're going to be hearing about that from Professor Uri Hassan later today if you choose to hang out with him. There's also a lot of strength in the development of technologies to probe brain function. And this is one of the co-directors of our Neuroscience Institute, David Tank. He is on the advisory council to the President's Brain Initiative. And I just wanted to point it out, you may see him around the building today, but David Tank is our co-director, and there's been a lot of activity at the national level to start developing technologies, a lot of interest at the national level to develop technologies to probe brain function. And I do want to talk with you about that today and show you some of the things that are possible with the new technologies. Because I think that initiative is not, Washington is not leading the scientists. What's been happening is that for the last 10 or 20 years, there's been tremendous progress by neuroscientists and by biophysicists and other researchers to develop ways of looking into brain circuitry or to perturb brain circuitry, to get brain circuitry to do what we want or to watch what it's doing. And the big goal is to develop technologies to do things like potentially watch what every neuron in the brain is doing at any given moment, and to be able to understand principles of how the brain works. So the Brain Initiative is, getting to be a big thing. This year, there are something like about 50 federal grants from the National Institutes of Health and the NSF, the National Science Foundation, to go to investigators to work on the Brain Initiative. And Princeton actually has just been awarded 2 of those. My colleague Mala Murthy and Josh Shavitz and Bill Bialik just got one from the National Science Foundation. I just talked to Mala, and she's going to the White House next Tuesday to join other scientists to celebrate their part in that. And then David Tank and Carlos Brody, Ilana Witten, and Sebastian Sung and I, and again, Bill Bialik, we just were awarded a pretty large grant from the National Institutes of Health. And in November, we're going to be going to NIH to talk about that. And it's a huge honor, and Princeton is right in the middle of it. Okay, so here's my topic today. So I want to talk first by talking about the kind of thing we're trying to study in the brain. I'll show you a few technologies that that have been developed for studying brain function. And I want to show you how my laboratory has developed an interest in applying these technologies to looking at brain circuits in action, and also some projects that we have going on in the lab to understand a pretty wide range of functions that may sound different, but in fact, I want to argue to you today that these two things, human individuality and autism, we believe are actually, in some sense, pretty deeply related. And I want to show you how we're thinking about that. Okay, so that's where I'd like to go today. This picture, by the way, is just to demonstrate the complexity of the thing we're studying. The human brain has about 100 billion cells that make electrical signals. Those cells are called neurons. And this is one neuron. And this is a neuron that I filled with fluorescent dye. And you can see that it's a microscope image. And this whole thing is about 1/10 of a millimeter wide. And this is a pipette that I brought in. I filled the cell body of the neuron. And you can see this elaborate, Baroque, almost Rococo, I love saying Rococo, is Rococo structure. That's the dendritic arbor where all the inputs come in. I say Rococo every chance I get.
Is it actually square, or is that an artifact of your methods?
Most of them are kind of elliptical, but what happened here is that this is actually in a slice of brain tissue, and when you slice the brain tissue, you can't slice the dendritic arbor perfectly, and so one of these edges is probably a blade cut, and the other edges are real, okay? So yeah, what is actually accurate about this, you can't quite tell from looking at this image, but this structure gets about 100,000 inputs, and it's several hundred microns wide, and if you were to turn it sideways and look at it, it's only about 10 microns thick. And so this arbor is almost perfectly planar. And what's, like a what?
Like a C-fan?
Yeah, when Ramon y Cajal, one of the founders of neuroscience, first described it working in a laboratory in Spain, he said it looked like the espaliered trees that you can see in France. That was his thought about what it looked like. But it's really a very striking structure. I mean, I should just say that this is not all neurons in the brain look like this. This is actually a highly elaborate dendrite. It's the most elaborate known dendrite in the mammalian brain. It's in a part of the brain that I'm going to be talking about today, the cerebellum. And so it seems to have the job of collecting an unusually large amount of information, even by brain standards, an enormous amount of information, and funneling it down all through, imagine this whole thing is kind of a funnel that all goes in here, and then the output goes on this little windy structure, which is called an axon. And so this is an integrative structure that takes a lot of information, as it turns out, sensory information, motor information, and then sends it out on this little axon. And so actually I'm going to be talking about this a fair bit later on. Okay, so anything else about this? I actually love this image, so I'm always happy to talk about it. And those little nubby things, you can see little itty bitty nubby things, those are dendritic spines, and each one of those nubby things is an input to this neuron. So that's just to give you an idea of the scale of how much information. And so just imagine this kind of process repeated 100 billion times, and that's what we're talking about in terms of what brains are doing. And one of the central challenges in neuroscience in the coming decades is to understand how structures like this come together to generate thought processes. Yeah?
Since, to differentiate, let's say a visual cue from an auditory, would the dendrites be, in effect, intertwined? In other words, because it looks like that's one neuron.
How do pieces of information get distinguished? Right.
And wouldn't that are there other dendrites interweaved with that, but it's only visualizing it because of the injection of the dye?
This is a great question. Okay, so let me see if I can unpack this in a way that A, makes sense, and B, fits with what I'm going to say later. a general way to think about brain function is that even though there are, as I said, 100 billion neurons and each one's getting thousands of inputs, there are pretty elaborate and well-organized rules that brains use to establish their connection. And so one way to answer your question is that if you look at the periphery, so for instance, if you start, say, at the nose or the ear and follow those pathways as they go in, those are pretty precise maps. And so there are olfactory receptors in the nose, they all send their axons in, and they sort themselves out. And all the axons that correspond with a very specific component of odor all land in the same place in the olfactory bulb. And so that's a organizing principle that's known as labeled lines, where it's just like an old-time switchboard where there's a specific line and you know that it's your friend Dan or whoever it might be on the phone or whatever it might be the case. And so those are labeled lines where it is known exactly what's going on. And when you look at sensory maps coming into the brain, those lines are very well organized. The interior of the brain also has labeled lines. So for instance, just to come up with an abstraction, the feeling of reward, when you feel happy that you saw an old friend for the first time in a while, which may have happened to you very recently since we're all together, that feeling of reward is conveyed by labeled lines that reside in a place in the brain called the ventral tegmental area and secrete a chemical dopamine. And that dopaminergic reward is an example of, in some sense, a labeled line that's not a specific odor or sight or whatever, but the feeling of an unexpected positive event. So the brain is filled with labeled lines. Now, at the same time, I think what you're putting your finger on is that, just to unpack what you're saying a little bit more, some of these inputs are going to be auditory or, depending on where this is in the cerebellum, or visual or what have you, and they're all coming together and they're, right? And so there's a few answers to that. One is that in order to extract this information, the brain has to start mixing information, right? Because it's not like, your old friend always smells the same way. I mean, I'm not saying anything about your friends, but
Cologne.
right, yeah, exactly. Okay, thank you for saving me. All right, but it's not like that, right? Like we're constantly integrating information and making sense out of it. And so there has to be that mixing of information. So that's one thing. The other thing is that these connections are not always the same strength, and so they get stronger and weaker. And that has to be true because we learn from our environment. And so the brain has wiring that looks like this that mostly stays the same in adult brains, but is constantly changing in how strong and how effective it is. And that's a phenomenon that neuroscientists call synaptic plasticity. And so this idea of labeled lines becomes a fairly complex subject, and it turns into a question of, well, how can you tell, how do we learn language? Where does that come from? Because it's not possible to imagine a labeled line concept somehow fitting with language. And so there has to be mixing of information that happens, and that happens, there's layer after layer of processing in brains. And I think a big part of what neuroscientists are trying to figure out is how to take all these details that I just told you about and turn that into a synthetic understanding of how is it that we decide that that arc of light is mom smiling at us and not leaves, sunlight coming through the trees, that kind of thing. And so there's a lot that has to happen there. And so in some sense, we're getting into what neuroscience is trying to get done. And also actually what, I should say, since we're now sharing a building with psychology, phenomena that those men and women knew about 100 years ago and have been very interested in. And I view neuroscience as taking those ideas. I mean, they thought of a lot of the good questions and turning those into biological insights into how we become that, right? Where they might be interested in language or memory. And a neuroscientist might turn that into neural processing in hippocampus or in auditory cortex, or for that matter, Alzheimer's disease or Parkinson's, domains where psychologists have relatively little to say. So anyway, so sorry to go off about it, but does that, you had no idea that question was going to lead to that. All right. Okay, so let me talk just a little bit about how fantastically complex this thing is. I've already given you a feeling for it, but just to give you a sense for it, this thing that's about 2.5 or 3 pounds, give or take, or 1.2 kilograms, is a biological organ. And even though we care about it for the fact that it produces all our experiences, all our mental phenomena, love, hate, boredom, hobbies, whatever we like about our brains. This thing is a biological organ, and one of the most amazing things about it is that it only uses about 15 watts of power. So think about your body's entire energy budget as being 70 watts, and so you use about as much power as an old-fashioned incandescent bulb. And of that power, 15 watts goes to power your brain. And so that's actually, I mean, it's a lot of energy on the scale of biological organs, but it's remarkably efficient. So this laptop that I'm using right now to project this, when it's idling, it uses about that much power. And it is using about twice as much power as that right now. And so each of us, whether you're thinking or idling or sleeping or yelling or out on the playing field or what have you, your brain is always using about 15 watts of power. And in some sense, so that's about as much energy as the light bulb in a refrigerator. So I guess that makes us all of us dim bulbs. And every one of us, no exceptions. Anyway, you're always using that 15 watts, and so you may as well do something good with it. Okay, so I've already kind of outlined what I wanted to talk about today. I wanted to basically connect the idea that there are things that I think all of us care about in ourselves and those who are close to us. We all care about personalities, our own memory, our sense of identity and who we are. We care about all the functions, some of which I mentioned. And we also care about dysfunctions. If you or someone you know has any issue that you're concerned about, whether it's neuropsychiatric or whatever it might be, dysfunctions are also of great interest to us. And so these are the kind of things that we care about in everyday life. But what I would say, where I want to go today is I want to talk about these phenomena in terms of brain mechanisms and the kinds of things that we care about as neuroscientists and also in psychology, sort of intermediate on the way to this, are things like genes, right? Genetic inheritance that we get from our biological parents, developmental mechanisms that proceed over time, and experience and environment. And these kinds of mechanisms work through biological circuitry, and I haven't even put up here words like neuron and synapse. These kinds of biological processes are the ones that shape all these things that we care so much about. And so in neuroscience, what we're very interested in is trying to understand how one gets from here to this 3-pound organ to all these other things. And we do this not only in humans, that's the preferred organism of choice over in psychology, but in my laboratory and others in this building, we're also very interested in other animals. For instance, this is a human brain here. This is a rodent brain shown in cross-section. And this is a slide just to remind you that mammalian brains have a lot of the same components no matter what the species. And so if you look here in this rodent, this little dude has a cerebellum. We have a cerebellum. It has a neocortex, so do we. And the neocortex in us looks quite folded. This one looks quite smooth. And there's all these other brain regions. And so what that means is that because of what Darwin called common descent, we have a lot of the same biology and a lot of the same circuitry. And it's expressed differently. It grows to a different size. But the idea is by studying organisms like rats, it's possible to understand, generally speaking, what brains do, and eventually start linking up different levels of function and applying that knowledge to understanding, say, what humans do. So these 2 Brain Initiative grants that I mentioned to you, one of them is to me and Tank and Brody and Witten and Sung and Bialik. And that's a grant that actually is focused mostly on working on rodents. And the other grant I told you about, where Mala Murthy and Josh Shavitz and Bill Bialik, again, the one that they have, is actually focused on working on fruit flies, which you might imagine is pretty distant from humans biologically, but there are deep principles about how brain circuits compute that they're hoping to learn from that. So there's this commonality between brains that we're all trying to work on. Okay, so let me just talk briefly about technologies. And this is a little bit of a busy slide, but this is just to show you that there's a pretty wide range of technologies that are now available to start probing brain function. And many of these have come into wide use just in the last 10 or 20 years. So one example I'm going to be showing you some data from is a technology for a kind of microscopy that lets us look deep into brain tissue. I think in the news, everyone has seen MRI scans, CAT scans, functional MRI. Those give resolution of about 1 millimeter. But I just showed you a neuron that's 1/10 of a millimeter. And it had structures that are much smaller than that, 1/10,000th of a millimeter. And so it's necessary to develop technologies to look at those very tiny structures. And in several laboratories here in this building, including mine, we use two-photon microscopy, which is a kind of laser microscopy that uses quantum effects to examine single dendritic spines and single neurons as they process. And I'll show you some movies of that. In the Tank laboratory, they've pioneered head-fixed recording, where you can take an animal and train it to not mind being restrained, and to head restrain it, and you can actually use this microscope then to look and watch brain circuitry in action as this animal does some task. So those are examples of physical technologies. I'll show you that in a second. And then there are a whole bunch of technologies developed using molecular biology, using fluorescent proteins that originally were discovered in jellyfish. So green fluorescent protein comes from jellyfish. And what we as a field have done is develop probes to make these fluorescent proteins get brighter when a nerve cell is active. And so it becomes possible to spy on neural activity. And it even is possible using something called optogenetics to have other proteins that when light hits them, they generate electrical activity in a neuron. So now we can start using light not only to watch brain tissue in action, but actually to get it to do something. And so what that means is that we have new abilities to start playing a brain circuit the way that one might play a piano or an organ or a stringed instrument. Okay, so this is just to show you, is that okay? I kind of zipped through a few technologies there. So let me just show you the kind of thing that's possible. Now this is not two-photon microscopy, but this is just to show you, let's see, actually, this is probably not the right thing to show you first, but let me just show you this anyway. So this is the surface. It looks a little bit blurry, and it looks like some kind of like, I don't know, Apollo mission image from 1968 or something like that. But this is about 0.5 millimeter wide. And this is taken using a regular fluorescent microscope. And what that means is that this is an image taken using a microscope that you could get hold of pretty easily in a, say, high school science laboratory. And what we're looking at is the brain surface, and the reason it looks bright is that it's actually giving off light. And the reason for that is that what we in the laboratory have done is we've gotten this circuit, this piece of brain One of those is one of those rococo structures, rococo. And what we're watching is we're looking in the brain of an awake mouse on a treadmill. And at that particular moment when everything flashes, it's getting a sensory stimulus. And so I'm just trying to give you a sense that it is possible to watch brain circuitry in action, okay? So that kind of thing is possible. This is just with a conventional microscope. And I mainly show this just to show that the technology is sufficiently far along that it's possible to do these things with actually not such expensive equipment. And I've got this crazy bee in my bonnet lately to apply this kind of thing to K through 12 education because it should be possible. I mean, these things are within reach of the budget of a typical public high school.
Only if they misbehave.
So I have a 7-year-old. And my experiments with her are generally in the domain of observational. But we can certainly talk about that. She's quite a trip, actually. Okay, so let me just, let me see where I am here. Let me just show you at the other end of things, just to give you a sense of this, we're also very interested in watching these animals behave. And so we do things like this where we observe behavior, and we can actually observe animals from all angles. And what we're doing here is we're developing technology to record from these animals' brains while they're doing something, whether it's just simple walking or whether they're exploring their environment. And you can see from this animal's facial expression, it doesn't seem all that upset about what it's doing. It's just kind of hanging out. And so, so this is a kind of thing that we're doing in the laboratory, right? Head-fixed recording from rodents. Okay, so another kind of thing that's possible is that it has even become possible. Now, this is not living tissue. This is now preserved tissue. It's even possible to take brain tissue and render it completely transparent. And so you can't quite tell, but this is actually a photograph of a brain that we have cleared in the laboratory using a technology called Clarity. And the Clarity is an acronym that I won't spell out in full, but basically it's a way of getting all the lipid molecules, all the fat molecules out of a piece of brain tissue, and what's left is protein molecules, and it turns out that what's left is quite transparent. And this is a technology developed at that place with a good football team where in addition to playing football, they also do a pretty good job of developing technologies like this, and so there are our sisters and rivals. And it's possible using technologies like Clarity to trace connections through brain tissue from one region to another distant brain region. So when we talked about complex information processing, one of the things that's really important is to be able to trace information as it goes through the brain and to understand those long distance connections. And so a tissue clearing technology that makes brains transparent makes it far easier for us to reconstruct entire volumes of brain tissue and understand those long distance connections. So that's a technology that's super important to us. And also I should say that Sebastian Sung, who we just hired recently, who we recruited from MIT, is very interested in brain circuit reconstruction and is actually coming up with some pretty interesting insights into brain function from his reconstructions of brain circuitry. Okay, so those are examples of the technologies we're using. Now I want to get into the question of what kinds of questions that we're starting to investigate, mostly in my laboratory. That's what I'll be talking about today. Are we okay? Any questions about the technology? Because I'm going to kind of move away from the tech and get into the biological questions. Okay. Okay, so now I want to talk about child development. As I mentioned, I got a 7-year-old. I would say up until about 5 years ago, I was mainly interested in learning mechanisms in the adult brain. And something happened in me that got me more interested in development. And I would not necessarily say that it was the act of parenthood, but I will say that that I was really struck. I remember going to the bookstore. Like I'm like any good academic. When my wife and I discovered that we were expecting, we waited till the last minute, and then shortly before our daughter's arrival, we started cramming. Good academic practice. So I go to the bookstore, and I see all these books like Healthy Sleep Habits, Healthy Child, and The Happiest Kid on the Block, and all these other books. And the thing I discovered is that in some sense, many of these books are about brain development, right? But it turns out that very few of them actually get into brain mechanisms at all. And so this book that you heard about before, I actually wrote another book called Welcome to Your Child's Brain, which is all about the brain mechanisms of how children's brains develop. And so I've developed a real interest in child development, both from the point of view of being a parent and also a neuroscientist. So what I want to show you in the last few slides, and then I hope that we can have a good conversation about it, is I want to talk about growing brains and how the brain is a physical organ, but it's got this interesting quality of shaping itself, right? We inherit genetic material from our biological parents. We have a genetic program that does a pretty good job of mostly taking care of itself and just needs normal loving experience to grow. But external experience plays a pretty critical role. And so the growing brain is this physical organ that goes according to its own agenda and uses experience, and then uses that to shape itself. And so this cartoon that I'm showing you here is meant to illustrate the idea that somehow experiences cause the brain to, sort of carve itself out in some sense. And I want to illustrate that by talking about something that is actually an example of that carving process that goes off track. And the fact that it goes off track is itself a demonstration of how amazing this mechanism is, because if it goes just a little bit off track, we start getting phenomena that seem pretty odd to us as human beings. But if you stop and think about it, they're actually a reflection of the biology that makes us who we are. So I'm going to talk a little bit about autism. I think many of you may already be aware that currently autism is diagnosed in 1 out of 68 small children in the United States. And so it's become widely recognized as a common phenomenon in small children. It's been recognized in ways that it was not recognized even 20 years ago. And so let me just talk about autism as a phenomenon of aloneness and sameness, where autistic children are socially isolated. They don't have a clear model of what it means to interact with other people. There are a lot of difficulties that they experience in interacting socially. And so think of that as an aloneness. And there's also a sameness that they prefer, right? So autistic children like things to be the same all the time, much more so than the rest of us. And so there's something called perseveration, where they just do things the same way over and over again. Maybe you have a 1 or a 2-year-old and you discover that he really only likes one toy, or gets super upset when you take a different route to preschool, or whatever it might be. And that's the moment when you notice, this kid likes things to be the same more than other children do. And so these qualities, when we find them in extreme enough levels, are called autism. So now, some of you might think that autism is on the rise, but I just wanted to point out something kind of interesting, which is that although the diagnosis rate of autism has gone up, and this is kind of an old slide, so it ends a while ago, other developmental disorders, such as mental retardation, have actually gone down, or actually that's learning disability. So learning disability here, mental retardation, these things have gone down. And it appears to be the case that what's really going on is that autism is not necessarily going up in the population, but it's more that we're taking children who have different qualities and we're calling them different things, in part, I think, because of the availability of therapies for autism that were not previously available, say, 20 years ago. And so this is a phenomenon that's known as diagnostic substitution. But anyway, whatever we call it, autism is a pretty interesting thing that, that really demands greater understanding. Let's see, so 9:37, okay. Let me just talk a little bit about both autism and other things and individuality. I promised you that I was going to link these two ideas up. There's a quantity that geneticists use called heritability, which can go anywhere from 0 to 1, where if heritability is 1, it's totally predicted by genes. And if it's 0, genes play no role in predicting whether something, you know, some quality of our bodies or our brains. The quintessential heritable feature is height. So if your father is above average height and your mother is above average height, it's possible to say with a lot of confidence that you are likely to be above average height for your sex. And so that feature of heritability is quite high for height, and so it's got a heritability that's close to a perfect 1, which is 0.85. An example of something that has no known heritability is acne, right? So that's been looked at, actually. And so therefore, whatever our children can blame us for, that would not be among them. Okay, so, but look at all these things in between. And it turns out that all kinds of things are in between. So geneticists have put in a huge amount of effort into studying disorders like autism, bipolar syndrome, depression, substance abuse. I haven't even shown schizophrenia on here. Most neuropsychiatric disorders, not most, but yeah, most of them actually, have some pretty strong heritable component that's not quite as strong as height, but is pretty strong. But you see here, autism actually has a pretty strong heritable component, and what that means is that it's probably caused by, ultimately, by combinations of genes. And then nestled in between these disorders, you can see these things that are normal traits, numerical ability and verbal ability, and they also have high heritability. And so what this suggests is a picture in which whatever happens to us in life, experience, nurturing, right, all the things, nutrition, prenatal experience, and so on, all those things have effects on what our brains become. But we start with some inheritance, and that inheritance is a starting point for where we end up. So it's not that genes are fate, it's that genes establish a starting point, and that starting point sets the stage for all the things that happen to us later on, right? And so the main point I want to impress upon you here is that this feature of autism is heritable, and it's heritable just like other things. There are other things I haven't put on here. For instance, personality is also heritable. So, and what that means is that you have personality traits that are like your parents, even if you're adopted and have never met your biological parents. You still have personality traits that are like your biological parents. And so a wide variety of brain traits, behavioral traits, actually have strong inheritance. Is that okay with people? Yes?
It just seems from people I know or clients of mine that the usual case is the two parents have no autistic traits and the child does.
That is true.
So, but
how could it be heritable and how could that be true?
Yeah, I mean, to a large degree, yeah.
Yes. You have seen my slides. Okay.
Okay, I confess.
All right, so let me talk about that, because actually the question you just asked me is in fact the key to what it is that connects individuality with autism. And let me just unpack that a little bit, okay? So geneticists have been pretty hard at work at finding specific locations on the chromosome, specific genes that increase the risk of autism. And by now, hundreds of them have been found. I forget, it's like between 300 and 500 have been found. And the general thought is that it's going to be in that. Actually, no, it's about 200 have been found. I think the estimate is that there's going to be something like 500 or more. So our chromosomes, our genomes are littered with these genes. But the weird thing about this is that these genes are actually quite common. So it's not that one person has an autism gene. Okay, so what turns out to be the case, this is a collaboration involving hundreds of researchers who I couldn't even put on this slide, and so I had to truncate it in odd ways. I was visiting a place, and every line of the author list where somebody at that place had an entry on the author list, I put their line on there because I wanted to make everybody I was visiting happy. And so there's some excellent people who I know who are on this list. I collaborate with this woman. Anyway, so the idea here in this title is that these genes are actually quite common. So most of us in this room are carrying some risk gene for autism or depression or bipolar or substance abuse, even if we ourselves do not have that. Even if nobody in our family has any of those problems, we are carrying these genes that have been demonstrated to increase the probability of this disorder. And so that's really, if you stop and think about it, that's kind of amazing, because it means that what it means is that we are walking around these alleles, these genes are present in our genome and have been around. And I mean, just to point out a fairly obvious statement, these genes have names, and so they have names like TSC1 and SHANK3 and DISC1. These are proteins that have jobs. We have to have these for our brains to function. We can't do without them. And so if we didn't have them at all, we would not, in many cases, there would not be a functional brain that came out of it. And so they're necessary proteins, but versions of them seem to increase the risk for disorder. So what's going on here? And now let me connect the dots a little bit here. This is now something I did, actually, speaking of human experimentation, this is something I did with the class of 2014. And when they first arrived, there's a thing called the freshman address, and I was invited to do that. And so it turns out that when incoming freshmen arrive, you may remember this from long ago, they're excited about being here, and they'll do anything a professor says. So before giving the freshman address, I emailed them all, and I said, hey, you're going to be here. I'm looking forward to meeting you in person. Before you get here, would you take a few minutes to fill out a brief survey? And I got a 90% response on the survey. Survey researchers never get 90% response on their surveys, but I got 90%. It's the only time in my life I will ever have that kind of response rate. And I asked them things like, well, what do you think you're going to major in? What part of the country are you from? And oh, by the way, do you have any neuropsychiatric disorders in your family among? Just by the way. And I found something. It's like a BuzzFeed article. What we found might surprise you, and then you have to click on it. So I will click on it now. All right. And so the first thing I found was actually not all that notable, which is that compared with the general incidence of autism in the general population of about 1 in 100, now somewhat higher, siblings of humanities and social science majors didn't report any notable difference. So on average, humanities and social science majors, I also asked them how many siblings they had. And when you considered how many siblings they had, this number is not any different from what one would expect from a general sample taken from the entire US population. But this is what I found. What we found might surprise you. So this is what I found. So siblings of science, math, and engineering majors have about a 3-fold higher rate of autistic siblings. And I think this gets to your question a little bit more directly. And in fact, this was of super great interest to me because I was a physics major in college. And in fact, my sister, as it turns out, is autistic. And so I am little green dude here, as well as the 1 in 25 Princeton freshman who I surveyed. So now let me just connect the dots a little bit here. So we've got a bunch of genes running around in our genomes that do important jobs in all of us. Common genes cause autism. I share half my genome with my sister. So what that suggests is the possibility that there are genes that are common that in most people don't cause autism, but might cause other traits that are, I would say, mostly considered normal, like an interest in science, math, or engineering. Okay, you might not think it's normal, but it's certainly within the normal range. And so the idea here is that maybe there are genes that cause individual diversity right, that make us predisposed to like different things. If any of you have multiple children, you may have noticed that even though you may do your best to treat the children equally and similarly, they might have very different personalities. One might really like soccer. Another one might really like painting or dance or whatever it might be. And so children from a very early age develop preferences as to what they like. And what that suggests is that there's some predisposition that we inherit from our parents. So imagine genes as now being like a deck of cards, and you shuffle the deck from mom and dad, and then you deal out the cards. And each kid gets dealt from the same deck. And I got a handful of genes that led me to be interested in physics and neuroscience, and my sister got some of those same genes, and for her it turned into a predisposition for autism. This is more generally true, and this is now a collected set of data from all the class of 2014. Now, you can actually give yourself this quiz. If you think about your first-degree relatives and you think about adding up a score, first-degree relatives, if you have autism in a first-degree relative, give yourself a score of -1. If you have depression in a first-degree relative, +1. So these are blood relatives we're talking about here. Bipolar, +1, substance abuse, +1. And now you've got a score that goes from -1 to +3. And if you look here, mostly people who are -1, who have this in their families but not this, mostly in the freshman class that I surveyed, they're mostly natural science majors. And here, at the other end, if they have 2 of these and not this, or all 4 of these, which is would be quite a feat, but if you have 2 of these and that gives you a score of +2, then you're more likely to be this. And the odds ratio here is a whole factor of about 20-fold different. So the odds of humanities to science are shifted by 20-fold going from here to here. And by now, I hope I've given you enough time to add up that score for yourself. And then think about what major you are. And the odds are that you are in one of the large rectangles. Okay? Does that make sense? Okay. So now I will say that so far, I've given you a hypothesis that's purely based on survey data. No molecular genetics, nothing like that. But let me just show you what we're doing. I have an undergraduate project. So I have, roughly speaking, a project that's done by graduate students and postdocs, and I've shown you some of the technological things we're doing with that. And I have another project that's mostly done by undergraduates. And the undergraduate-oriented project is one in which we're basically applying computational methods to start looking for genes that predict normal range traits. And we're looking in a database of people whose genetic information is already on file, and all we have to do is recontact those people. And the first community that we've contacted is one that's run by the Simons Foundation. Jim Simons has founded an organization that funds autism research, and they're based in New York City, and they have a community of families who participate in autism research, and we recontacted them to ask them questions. Now, also in that database, there's genetic information, and there's also personality information. And so what we've been doing is looking at the personality information on file and using big data methods such as machine learning to try to start looking for connections between known personality traits and genetic content. And so everything I've told you before suggests that normal range traits such as intellectual interests, maybe personality traits too, it suggests that they will have specific genes. And so the idea would be that we can find a specific gene that connects with a particular trait. So this is something they already had on file, and so this is our first pass to find out whether there's any merit to this idea. There's something called the broad autism phenotype, and what this means is it's other features that are like autism, but not quite pathological. And so it's a questionnaire that everyone can take, and it's got questions like this. I like being around other people, very rarely, very often. I'd rather talk to people to get information than to socialize. Well, of course, who wouldn't? Wait. People ask me to repeat things I've said because they don't understand. People ask me to repeat things I've said because they don't understand. When I make conversations to be polite, I can tell when someone is not interested in what I'm saying. Okay, so at this point in this questionnaire, I started to blush. I just felt this hot flush of embarrassment because I started realizing that I, in fact, could say yes to a lot of these things. I lose track of my original point when we're talking to people. I've slowly learned to, anyway. So if you go down this, you can add up a score. And it's possible to define a phenotype of people who are within the normal range who yet score high on domains like aloofness or difficulties with everyday pragmatic communication or wanting things to be the same all the time. And there's a phenotype, a characteristic called the broad autism phenotype. And it turns out that in the general population of parents, 5 to 9% of people have it. But in parents of autistic kids, 14 to 23%, right? So that's something like 2 or 3 times as high. And so what that suggests is, again, the possibility that in this case, personality might somehow be co-inherited, right? Have common features with autism. And again, a possibility is that it could be genetic. Okay, so now we get to some actual human genetics. Kate Shulgina in my group has been looking in the genomic information that's available at the Science Foundation and looked to see whether there are genes that occur more often than you would expect by chance among people who have these traits of rigidity, of aloofness, of difficulties with pragmatic communication. And obviously these letter codes will not mean anything to you, but anything that's color-coded, yellow means that it's known to be a gene whose variation is linked with intellectual disability or autism. Orange means that it's specifically linked with this disorder that's autism-like, Fragile X, mental retardation. It shares features with autism, or genes that are expressed in synaptic connections. And so you can see here that there are actually an awful lot of genes. And although there's some statistical questions that we have to get into, this is the first step in a chain of discovery in which what we're hoping is to use this as a starting point to start looking for more and more evidence that there are genes that both cause individual variation in all of us and also dysfunction in those that we care about. Is that okay? Okay, I'm almost done here. Let me just show you one last thing. And I've actually totally broken my promise. I said I was going to be done by 9:45. Total lie. But let me just talk a very little bit about something we're working on right now that we're super excited about. I wrote a piece in the New York Times earlier this year. I think it came out in February, I believe. Was it this year? Yeah, I think it was, or maybe it was last year. I lose track of time. But anyway, I had a piece in the New York Times about risk factors for autism. And we often hear in the news about things like vaccination, okay, which has is associated with no increase in risk, no change in risk relative to the general population. We also hear about things like parental age. And so when we are bothered by our parents about getting married and having kids, or perhaps when we ourselves bother our children about getting married and having kids, these things attract obvious interest, but that's a pretty modest increase in autism risk. But there are other things that we don't hear about so much. And these are environmental events, like having a cesarean section when it's not medically recommended, or very stressful events, such as leaving your home country during pregnancy. Your baby is 2.3 times more likely to be on the spectrum. Fleeing a hurricane strike zone. PTSD is another one, right? So these are all events that happen prenatally, environmental events. Prematurity. Here's one that's postnatal, which is that being put into certain orphanages where there's just no social input that's given. These kids get just fed and changed, that's all they get. And so that's an example of an environmental event that happens after birth. But here's what I really wanted to talk about next in the last couple of minutes. Sharing half your genome with somebody with autism, or sharing all your genome with somebody with autism, carries a tremendous risk of autism, which is the demonstration of the thing I was saying before, which is that autism is mostly genetic. But in the last few minutes, I want to talk with you about just this last thing in here. This is one environmental event that's kind of odd, which is injury to the brain region back here, the cerebellum, which is a region that my laboratory studies. And injury to this region carries a risk of autism just as great as genetic inheritance. And so even though over there on the left, media coverage is what we hear about in the news, over here on the right, from a scientific standpoint, is where all the action is. And so, roughly speaking, what we're interested in is the idea that somehow internal communication in the brain, whether it's driven by genetics or by injury, that genetic program might act through the cerebellum to drive brain development off track. So it's not that all autistic kids have cerebellar damage. It's that this particular statistic is telling us something about the biology of how brains develop, and that maybe genes can also drive miscommunication between the cerebellum and the rest of the brain. And so what we're currently interested in right now is this idea that this brain region here in back, the cerebellum, communicates with other brain regions. And what we believe is that communication between the cerebellum and the neocortex, this very large part of our brain, we believe that that communication is critical in early life for setting up social abilities. And so we're extremely interested in testing that idea. And roughly speaking, the idea is to use the brain technologies the brain initiative technologies that I mentioned before, and to do something like, in early development, take this brain region out of the game and say, let's inactivate it in an animal while it's a pup, then let it grow up, and then let the brain region back in the game and say, did it play a critical role in guiding the function of some frontal region? And so this is something that we're very excited about right now, and in the coming couple of years, we're hoping to test. Yeah? Was there a question somewhere? Yes?
Do any of these studies differentiate by gender?
Yes, that is usually differentiated by. So I mean, there's a lot to say about autism and gender. The sex ratio of autism, I think, as you probably already know, is about 4 to 1 male to female. And so it seems that there is something about the biology of male brains that predisposes boys more likely to become autistic than girls. But every finding I've told you so far, it applies to both sexes. Yeah.
Are any of those, just similarly, any of the genes on sex, are they sex-linked genes?
No. Most of them are not
So
on the
it's
X or Y chromosome.
development to the brain.
Exactly. It is not, right. So the obvious thing to do is to look on the X or Y chromosome. That it turns out not to be the case. Yes?
Does injury to the cerebellum that you said was associated with autism in humans cause other types of disabilities or problems, or is it really specific?
Yeah, let me unpack that a little bit. So I didn't tell you the reason for why that's a surprising finding. So for centuries, the way that people have understood what a brain region might be doing is to see what happens when it's injured, right, in an animal or in a human. And the usual thing that you see in people with cerebellar injury is that they become clumsy. Their movements become what's called ballistic, where instead of just making a gentle movement to pick something else, they overshoot, right? So there's this inability to balance and so on. And so mostly what's observed in adult neurology is that kind of problem with balance. But the notable finding here is that specifically injury at birth to the cerebellum is associated with a large risk. And furthermore, I left out a whole bunch of details like it's not just any part of the cerebellum. It seems to be specific parts of the cerebellum, maybe the part near the midline, maybe the part that connects with the prefrontal cortex. And so there's some pretty specific findings that I sort of left out. But the general idea here is that there seems to be something special about the cerebellum because it's the only region that when you injure it early in life leads to something that looks like autism. But when you, like if one of us unfortunately got a cerebellar injury, we would not become autistic. And so specifically, we are very interested in the idea that it's playing some kind of teaching role in early life that it doesn't play in adulthood. Okay, so I'm sorry, I kind of got excited and I compressed all of that into about, blah, 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 we're going to inactivate it. Yeah, I think we're almost out of time. Let me just see what else. Okay, so just to end with the punchline, the idea that we're really excited about in testing in my laboratory in the coming years is to test the idea that the cerebellum may play a critical role in shaping the social neocortex, that it may build itself, but the brain may build itself, but it does so through internal communication during developmental periods that are known as sensitive periods. And I hope that during the precepts, if you meet Amanda or Alexandra, either Dr. Crocker or Dr. Badura will perhaps talk about sensitive periods just a little bit. And so where we're hoping to go is to understand how some network of neurons in the brain might play some critical role both in adult life and also in constructing the social brain. So why don't I stop there and take more questions. Thanks very much.
I just had a question going back to the, let's say the 6 personality traits. Of those traits, do you have heritability factors that are higher for some of those 6 major traits and lower in others, say openness or agreeableness? Will those have a higher number or a lower number?
Yeah, I didn't actually show that, but you're talking about, right, so things like the 5-factor model, and there's things like agreeability, extroversion, and so on. All of those traits have been shown to have some degree of heritability. They all get a little bit stronger as we get older, and so we turn into our parents, more or less. I don't know that any of them are more strongly heritable than others, although I think part of the difficulty there is that when you make a sort of an interview-based measurement, you're measuring a mixture of many different brain traits. And the kinds of things that we measure when we meet a person are not necessarily indicative of any particular brain region or any particular brain circuit. Sorry, I'm going to stop this because I can't, when this thing is playing, I can, no, but I'll play it for you later. But But I can't stop looking at it when I'm playing it, so it's distracting me down here. Right, so anyway, the point is that brain traits are complex, and so it might not necessarily be surprising that all those factors seem to be similarly heritable. I think if we started to understand, if we could start teasing apart the components a little bit better, and we started to understand brain circuit mechanisms, it might start looking a little bit different.
Is that trajectory going straight up, or does it have levels when you say we're coming more like our parents?
It's not straight up. So roughly speaking, to get to the literature a little bit, in our 20s, personality is moderately heritable, and then the heritability goes up to like 0.6 or so by the time we reach our 50s. And so, you know, we're not doomed to become our parents, but there is this unmistakable trend to become more similar to them with time.
Thank you.
And I'm following the microphone around, by the way, just in case you're wondering. Yes, over there. It is.
I have a son, Tommy, 22 years old, who's madly in love with Midori. How far is your field, Sam, from being able to counsel Tommy and Midori that if you get married, here's what's going to happen with a whole, not just autism, but a whole series of disorders. Maybe their children might be born blind, they might want to become lawyers, investment bankers, things like that. And secondly, how far are we from being able to manipulate the genome to say, Tommy and Midori, if you do like to get married, we have a way of dealing with and mitigating some of these risks?
Okay, Tommy and Midori. Dear Tommy and Midori, we're delighted to let you know that we've tested your embryo and we've discovered that of the major diseases that are determined by one gene, your embryo doesn't have any of those disease risk genes. However, those high-risk genes are only a tiny fraction of all the things that we know lead to disorders. And most of the genes that we know about that cause disorders only carry a low risk. And what that means, there are several consequences of this. One is that we actually have only discovered a small fraction of all the genes that increase the risk of disorders. The second is that so many traits are complex and caused by combinations of genes that it is very hard to predict what's going to be the outcome when Tommy's and Midori's genes are shuffled. And I think that it's pretty far. I honestly, this is going to sound a little bit inverse, but I'd say where the field is likely to head, at least in Tommy and Midori's lifetime, is that there's a lot of discovery of high-risk genes where the gene has a really large effect. That has come along pretty well. But this thing that I was talking about today with common variants is almost a step backwards in the sense of basically pointing out that those genes mainly cause normal range outcomes when expressed in combination. And what that means is that there's a certain fruitlessness to genetic counseling for a low-risk gene, because you might think that you'd like to get rid of the SHANK3 gene for autism, or you might think that you'd like to get rid of the DISC1 gene for schizophrenia. But actually those genes are doing pretty important jobs, and what you're also doing is doing an uncontrolled experiment on shaping who your child becomes. And so I think that my current take on it is that the kind of things that we're working on are kind of negative for genetic counseling, in the sense that one should actually be pretty careful about getting too ambitious in trying to select for outcomes. Now, you know, their children or grandchildren, I mean, one can imagine knowing more about these things. I mean, if our project succeeds, we will be able to give a teeny tiny amount of information about whether their kid is more likely to want to major in classics or physics, right? So that might happen in the future. But even then, that's not fate, right? So I think that I would characterize these discoveries as being more along the lines of discovering general truths about where we come from, but not necessarily applying those to doing something with a particular embryo.
I understand studies have been done of the brains of psychopaths that demonstrate abnormal or failure of certain connections between the frontal lobe and some other places in the brain. Does that have any relation to the kind of thing you're talking about? And could you comment on that?
Yes, so one way to start, so I've started by talking about genes, And at the other end, we get to brain function. There's an echo that's developing here. It's quite odd. Let me just, okay. Okay, anyway, I feel like if I move around, it'll go away. And so one way to make that connection is to start asking things like, how do genes specifically influence brain structure? And there are people who are working on that. One difficulty with those studies that you've been reading about in the news is that in no case are those studies as definitive as that they sound like in the news. So roughly speaking, an effect can be pretty small. As long as there's any effect at all, it's good enough to discover, publish, and get promoted or get a nice faculty position on. And it can be more than, as long as it's more than no effect, that's an interesting effect. But that's very different from looking at a brain and saying, ah, you are a psychopath. And so it turns out that's something that the statisticians call effect size. And the effect size of most of those kinds of things is actually not very large. It's quite small. And so it is often not very predictive in individuals, yet there's an effect when you look at large groups. And so, so I think that work is not nearly so advanced as one might be led to believe by reading the science pages. I mean, there's a real effect there, but it's actually pretty small. That having been said, it's probably frontal regions, for instance, that are a little bit disrupted in brains of psychopaths. I think there's a lot of interesting biology there. I just think it's a little bit less, sorry to rain on it, but it's just, the path is genes to brain structure, but the path is only beginning to be carved out. Yeah, there, and I think you raised your hand a few times, so maybe if you can go to him. Yeah, okay, sorry, over here, yes.
On your slide on the relative risks of autism,
yes.
The one I found most sort of counterintuitive was the elective cesarean. Is there some mechanism?
Here's the idea. I realized that I was running a bit short, and so there's a whole song and dance there's a whole biological story that I usually tell at that point. I think a good way to synthesize all those items on that bar graph is as follows. We inherit a genetic program. It goes through developmental stages in utero, and then in the second half of pregnancy, maybe in early postnatal life, somewhere in that time window, the developing brain seems to be sensitive to external events in ways that can take a brain that is probably going to be okay and then increase the risk to drive it into a range where a more diagnosable outcome occurs. And so the right way to think about prematurity is that the womb is a really nice place. And to be born 6 weeks prematurely is to be put into an environment that is very different from the prenatal environment. And that is stressful for the baby. So just for example, if autism is on the rise, which is not agreed on at all by epidemiologists, one possible source of that would be that, that in prematurity, premature babies survive better than they used to. And so kids who are at risk might survive now when they didn't survive 20 years ago. Sorry, you have a follow-up.
But my question wasn't about being premature, it was about elective cesarean.
Oh, I'm sorry, elective cesarean. Well, okay, right, so I'm sorry, I sort of got a little off track there. Elective cesarean. Yeah, elective cesarean is actually usually done like a month early, right? That kind of thing. I mean, you'd be amazed at how early, right? So my sister was an elective cesarean. Like the obstetrician in my sister's case, this is just anecdotal, but he wanted to go on vacation. And I mean, I can't really say whether that was causative because it's only one instance, but she was delivered a month early. Yes, and maybe, yeah.
On the genetic counseling, how do you see it playing out in the hereditary sense in view of the 23andMe, history?
Oh, well, as I was saying before, I think that high-risk genes are the low-hanging fruit of genetic discovery, and I think that's going to continue to be something that's super interesting. Predispositions for breast cancer, there are some famous Alzheimer's genes that are really high risk, and I think those are very important discoveries. But I actually think that a lot of that low-hanging fruit has been picked, and the more difficult problem is understanding how these genes combine with one another. And so, look, I'm carrying autism risk genes. I almost certainly am. And yet I am not autistic. And so there's an extreme difficulty in understanding how these genes talk to one another. And that is something that a naive gene chip approach, I think it's a little ways before that's going to happen because the statistical power is not really there. And I think it's going to be a fair time, it's going to be a while before that's possible. And I would actually question whether that's desirable. Anyway, sorry to be anti-technology on that front, but I know everyone wants to say, oh yeah, you're going to just spit on a chip and you're going to find out, no, that's not, or whatever. How much time do we have left? Yeah, how about you? And then I'll stick around afterwards, but
You mentioned that links between the cerebellum and neurocortex during development that are critical for autism. Is it possible to cut those links?
Yeah, I think a really interesting consequence of this is, let me just to connect the dots a little bit. In the study, I'm going to start with a very different place. In the study of Parkinson's disease, a major game changer was understanding that Parkinson's disease was not a general degeneration of the brain, but was a very specific degeneration of the substantia nigra, where there are neurons that make dopamine, and those neurons die. And the moment that that was discovered was a big moment in the treatment of Parkinson's disease. And so my general thought is that if it turns out to be true that cerebellar neocortical communication is a key type of internal brain communication that drives autism, now we have a brain structure that we can start saying, okay, well, you know what? The cerebellum might be a location for intervention. So maybe bad information from the cerebellum is adverse for neocortical development. Maybe it would be better to just take it out of the game entirely. Just put a little, I'll just make it up for a second. This is just me talking 50 years in the future. Maybe we discover that a child is not making eye contact or what have you, and at 1 year of age we say, maybe you should take the baby and just put this device on the back of the head for the next year and give intensive therapy to really teach her or him social interactions, and have this device that just takes the cerebellum out of the game, that just silences it while we're playing these games with the kid, to really get her really on track. And then at the end of it, we take the device away. And so one possible outcome of understanding the biology of what brain region goes off track is to now have a target for interventions. And I think a lot of interventions for depression, substance abuse, what have you, are things like pills that we take. And one of the huge difficulties is that that kind of intervention, in some ways, really hits a wall, because pill-based interventions just go throughout the brain. And once neuroscientists start being able to understand specifically what a brain region does, that starts turning into practical therapies. Is that the kind of thing you were thinking of? All right. Okay, so I should probably stop, and thank you all again. Thanks very much.
Thank you, Professor Wang, for a stimulating discussion this morning. I have a few housekeeping notes. You all have on your name tags, you have at the bottom, it'll tell you where you're going. So the first thing on the bottom line should say, but the top by your congregate in the lobby upstairs where you came in. And if you opted to participate in the morning assignment, should have been handed out to you on the little red or orange card. Next session begins at 10:45. There's coffee outside for those of you who haven't, like me, woken up yet. And again, thank you. Thank you we have a couple of class presidents in the audience, Susan Patton, 77, Van Williams, 65, who have gotten their classes together for mini reunions. 79's president is not here yet. He'll be here tomorrow. So thank you all, and enjoy the rest of your day. I hope you find it as stimulating as Sam's lecture.